Hello friends, welcome to BMH Learning. This video is about Amplicon sequencing. Amplicon sequencing is a method of targeted next-generation sequencing that enables you to analyze genetic variation in specific genomic regions. This method uses PCR to create sequences of DNA called amplicons. The method employs oligonucleotide probes designed to target and capture regions of interest, followed by next-generation sequencing. Amplicons from different samples can be multiplexed, also called indexed or pooled, which involves adding a barcode or index to samples so they can be identified. Before multiplexing, individual samples used for Amplicon sequencing must be transformed into libraries by adding adapters and enriching target regions via PCR amplification. The adapters allow formation of indexed amplicons and allow the amplicons to adhere to the flow cell for sequencing. Amplicon sequencing is typically used for variant detection. Amplifying your sequences of interest with a set of fusion primers is the method of choice. A fusion primer consists of a target-specific sequence at the 3' end and a fixed sequence at the 5' end for the sequencing approach. We can name them as primer A or primer B. For sequencing several samples in parallel, you may pool several barcoded samples. Barcoding of Amplicon samples is done by insertion of multiplex identifier tags or MIDs into fusion primers. As each Amplicon molecule within a mixture of amplicons can be sequenced individually, the technology has the power to detect rare variants with detection limits of 0.5% and even lower. This was all. Thanks for watching.